Diseases have plagued humans since man first walked on the earth. Three of the most lethal and incurable diseases are cancer, Alzheimer's disease, and heart disease. Cures for these diseases seem to be a lot more probable with the discovery of O-linked acetylglucosamine, or Oglicnac. Oglicnac is a type of carbohydrate that can be added to proteins to change their function and has been found to play a role in each of these diseases. Here, Dr. Michael Boyce of Duke University talks about Oglicnac's role in cancer.
Those proteins are known to be oncoproteins, meaning proteins that are overexpressed or inappropriately turned on in cancer. And some of those proteins are tumor suppressors, which are proteins that usually are turned off in cancer. So there's a lot of interest in the field in understanding how Oglicnac might regulate those types of proteins in cancer, how it might go wrong in cancer and contribute to cancer or metastasis.
Oglicnac also plays a role in heart disease. When it combines with the proteins in the heart, or the heart is oglicnosylated, the Oglicnac is removed during times of stress and has a protective effect on the heart. This implies that Oglicnac may play a role in the prevention of heart disease. Oglicnac also plays a role in Alzheimer's disease. Tau is a protein in the brain that deals with cognition and memory formation. Scientists have found that when tau is healthy, Oglicnac is attached to its surface, and when tau is damaged, for example in an Alzheimer's brain, the Oglicnac is no longer present. This implies that Oglicnac may play a large role in keeping the brain healthy and working correctly. Since Gerald Hart and Carmen Rosa Torres discovered Oglicnac, the major issue in the field has been to find methods that are able to effectively study Oglicnac in cells. Originally, mass spectrometry was attempted, but this type of spectrometry damages the cells, and so the Oglicnac is lost before it can be studied. Another method that was attempted was electrophoresis, which uses charges to separate different materials from one another. This method was not effective enough, as Oglicnac has very little charge. Two more effective methods emerged from the two that failed. The first is ion trap mass spectrometry, which is able to detect Oglicnac as it does not damage the cells. The other is known as tagging, and it uses modified proteins that attract Oglicnac. It is more effective because the proteins are easy to study, and so it is much easier to quantitatively study Oglicnac as they are attached to the modified proteins. Dr. Michael Boyce of Duke University uses a variation of this method in his own lab.
But basically it's a way of taking an unnatural sugar that you synthesize in the lab, that has an unnatural chemical group on it, something that you could trace somehow. And so if you feed it to the cells, if it's close enough to the natural sugar, then they almost kind of won't know the difference. So they'll take it up and they'll use it as if it were the natural sugar, and then they'll put it onto their glycoproteins. And therefore you can come in later and using a chemical reaction that will specifically react with that sugar, that's usually what we do, then you can detect those proteins, you can purify.
Boyce is the only individual studying Oglicnac at Duke, and this is the first time he is a PI. Boyce is currently studying
an intracellular form of glycosylation, meaning inside the cell, and that affects protein function. So most of the projects in my lab right now focus on that form of glycosylation.
Oglicnac may have a large impact on curing diseases and the world. This is why Oglicnac is at the cutting edge of science.